Welcome everyone. This is the lecture on the cell cycle. We're going to talk about mitosis, binary fission, and cancer today. Please make sure before you start listening to this lecture that you print off the lecture slides so that you can follow along and fill in the blanks as we go. Also, you'll be able to jot down any questions you have as we're working through the lecture. So our objectives for this lecture are to describe the events of the cell cycle, describe how mitotic cell division results in genetically identical daughter cells, and explain the cellular basis of cancer. So the cell cycle is the life of a cell from its formation until it divides. The function of cell division, or why do cells divide at all, is to reproduce, to grow, and also to repair tissues. And these are some pictures of cells dividing. All right, some terms you'll need to know. Genome is all of a cell's genetic information. So all of the chromosomes, all the genetic information, all the DNA, we call that the genome. In prokaryotes, it's a single circular chromosome. In eukaryotes, it's all of their linear chromosomes together make up the genome. For example, in humans, we have 46 chromosomes, so that makes up a human genome. A mouse genome only has 40 chromosomes, and a fruit fly genome has 8 chromosomes. So is chromosome number related to complexity? So it's an interesting question, and the answer is no. There are organisms that are much simpler than humans that have way more chromosomes than we do. So don't be misled and think that the more genetic information something has, the more complex the organism must be. All right. Before a cell can divide, each chromosome has to be copied or duplicated. Duplicated chromosomes we call sister chromatids, and they're connected or attached to each other by a centromere. It's this little pinch point. So after this first single chromosome copies itself, and you learned about DNA replication last week, we have a second sister chromatid here. So they're connected by the centromere. And then when the cells divide, they separate, and each daughter cell gets one of the sister chromatids. Here's a micrograph, a picture of a microscope from a microscope of a real pair of sister chromatids. All right, so let's talk about some different cell types here. Somatic cells are all the cells that make up your body. They're diploid, which means they have 2 copies of each type of chromosome, one from your mom, one from your dad. And we call that 2N. There's 2 of everything, 2 of each chromosome in a diploid cell. These cells divide by mitosis. So in humans, our diploid chromosome number is 46. It means you get 23 chromosomes from your mom and 23 chromosomes from your dad. Gametes are different. These are sex cells like sperm and egg. They're haploid. They have just N, or 1 copy of each chromosome. And they divide by a different process called meiosis, which we'll talk about in more detail in the future. So for humans, each gamete, each sperm, and each egg cell have just 23 chromosomes, which makes sense, right? Because when 2 gametes come together, then we'd have one 46 chromosome diploid zygote, the cell that will divide by mitosis to produce a human, or whatever the organism is. But if we're talking about these numbers, a human baby. All right, take a moment and describe the structure of eukaryotic chromosomes. When you're ready, let's move on. All right, so phases of the cell cycle. So the cell cycle, cells spend most of their lifetime in interphase. And this is when they're just doing the work of being whatever kind of cell they are. And we're talking about somatic cells for the rest of this lecture. We'll come back to gametes when we talk about meiosis later on. But for now, mitosis, we have mitosis just happened. There's 2 daughter cells. We're going to follow this daughter cell throughout her cell cycle. So first, she grows through a growth 1 phase. So this is when this cell is growing in size. It's doing the work. So if it's a skin cell, say, it's doing its job as a skin cell. Some cells stay in G1 and never move on. But say this one has the signal to move on and make a copy of its DNA. The S phase is where DNA is synthesized or replicated. There's another checkpoint here. If the cell gets the go-ahead to move on into the second growth phase, it's going to start making extra organelles so that when the cell divides, both daughter cells have all the organelles that they need. But this is all interphase, G1, S, and G2. This is where the cell spends most of its time just being a skin cell or whatever kind of cell it is. If it gets a signal to move into mitosis and divide, that's when this mitotic phase begins. There's mitosis as the cell divides its contents and chromosomes into 2 opposite sides of the cell, and the cell membrane begins to pinch off. After the nuclear membranes separate, that ends mitosis. When the cell membranes separate, that is the finish of cytokinesis. And at that point, we have 2 new daughter cells. And the whole cell cycle starts again. So the mitotic phase alternates with interphase. So cells are always either in interphase or mitosis. All right, so interphase is 90% of a cell's time, basically. In the growth 1 phase, this is where cells grow and carry out normal cell functions. S phase is where the chromosomes are duplicated. And G2 phase is where the cell's preparing for cell division by making extra organelles. The M phase, or the mitotic phase, is made up of mitosis, where the chromosomes and nucleus divide. And the last part of the mitotic phase is called cytokinesis. This is when the cytoplasm divides or the cell membranes separate. All right, let's do a quick comprehension check. So what's the longest part of the cell cycle and why? And next, if the diploid number is 46 chromosomes, what is the haploid number that you'd find in a gamete for that organism? All right, so let's talk about the phases of mitosis. I want you to, we're focusing on these 4. Depending on which resources you look at, you'll see some like in-betweener phases. We don't need to worry about that. We're focusing on prophase, metaphase, anaphase, and telophase. And a good way to remember that is PMAT. All right, so here's the G2 part of interphase. We have a cell with a complete nucleus. The chromatin is duplicated, so there's a lot of DNA inside of that nucleus, right? As this cell moves into prophase, the first part of mitosis, we see some things start to happen. First, these spindle fibers begin to separate as the centrosomes migrate to opposite sides of the cell. At the beginning, in an interphase, the two centrosomes with centrioles, these are just kind of hanging out together in one part of the cell. Another clue that we're entering prophase is that the DNA begins to change shape. So instead of being all loosey-goosey as chromatin, they begin to bundle up as chromosomes. And they should be paired with their sister chromatids. In metaphase, the next phase, we have our centrosomes at opposite poles of the cell, and these spindle fibers from those centrosomes are attaching to the chromatids, which are all lined up kind of along the equator of the cell. So if you see the chromosomes all lined up in the middle like this, then you know it has to be in metaphase. In anaphase, this is when those spindle fibers begin to retract back up into the centrosomes. And as those spindle fibers retract, they pull the sister chromatids apart to opposite sides of the cell. And now at this point we call them daughter chromosomes. The last phases are telophase and cytokinesis. Telophase is when the nuclear membranes begin to reform and they separate the nuclear contents. So we have two separate nuclei being formed. That's telophase. Cytokinesis is when we get this cleavage furrow and the cell membranes between these two new daughter cells begin to separate as well. So you got prophase, metaphase, anaphase, telophase, and cytokinesis. In these images, by the way, the DNA is dyed blue. The spindle fibers are stained green. All right, so mitosis is a continuous process with observable structural features. The chromosomes become visible in prophase. They align at the equator in metaphase. They separate in anaphase, the sister chromatids separate, and then we form two daughter cells in telophase and cytokinesis. All right. How about for this one, I should change the question. Why don't you define what sister chromatids are here? Good. And after you've done that, I'll tell you homologous chromosomes would be the same chromosome number, but from the other parent. For example, you have, like for chromosome number 3, you have one copy from mom and you have a copy from your dad. Those are homologous chromosomes. They're both chromosome number 3, but they're not sister chromatids. When your cells get ready to divide and that chromosome number 3 from mom duplicates, the two chromosome number 3s from mom are sister chromatids. And the two chromosome number 3s that have been duplicated, those are sister chromatids from dad. Homologous chromosomes are the same chromosome number, like 3, but one from mom and one from dad. They code for the same genes, but they have different information, right? All right. So here's a zoomed in picture of prophase. You can see the chromosomes are becoming visible and the spindle fibers and centrosomes are beginning to separate. In metaphase, the chromosomes line up at the equator and the centrosomes are at opposite sides of the cell with spindle fibers attached to each sister chromatid. In anaphase, the spindle fibers retract, separating the sister chromatids. In cytokinesis, the cytoplasm of the cell is divided. In animals, we call this a cleavage furrow. In plant cells, a cell plate forms. That's the beginnings of the cell wall for a plant cell. So here's a micrograph of a cleavage furrow in an animal cell. And here we can see a cell plate beginning to form, separating two new plant cells. All right, and here's pictures, and these are actually from onion root tip cells, but you can see the DNA stained this dark purple color. So we have prophase here where the chromatin is condensing, and they're beginning to form chromosomes. In metaphase, everything's lined up at the center of the cell. In anaphase, the chromosomes begin to pull apart. And in telophase, we see these two young cells beginning to separate from one another. So, what I'd like you to do is pause the video, and I want you to see if you can identify or circle and label on your printouts cells in each of the 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. See if you can find them all here. All right, comprehension check. So at the end of mitosis and cytokinesis, how do the daughter cells compare with their parent cell when it was in G1? And here I'm specifically talking about like the genetic information. Do they have the same DNA and the same chromosomes as their parent cell did at that point in its life? All right. So we've covered eukaryotic cell division by mitosis. Next, we're going to talk briefly about how prokaryotic cells divide. And prokaryotic cells divide by a process called binary fission. It's similar to mitosis, but much simpler. Because remember, prokaryotic cells have just one chromosome, and they lack a nucleus. So things are more straightforward. All right, so the first step is chromosome replication begins. And here we go. And this whole spaghetti-looking mess here is all one chromosome, right? So as it goes through and replicates, eventually when it's done, we'll have 2 copies of the same one circular chromosome here. Once the genetic material is done being replicated, it begins to separate to opposite sides of the cell. The cell membrane begins to close off, and 2 daughter cells result. All right, so back to eukaryotic cell cycles. We talked about interphase as everything except the mitotic phase. We said the first phase of interphase is growth 1. Then there's the synthesis phase where DNA is replicated, and then growth phase 2. Now, each of these red spots, these are called checkpoints. And a checkpoint is a control point where stop or go signals are regulated by the cell. So in the G1 checkpoint, this is the most important checkpoint. It's controlled by cell size, growth factors, and environmental factors. If the cell gets a go phase here, it will complete the whole cell cycle. If it gets a stop message here, the cell enters a non-dividing state called G0 or G0 phase. And it's just going to stay whatever kind of cell it is and do that cell's work, but it's not going to divide. Nerve and muscle cells stay at G0. Liver cells can actually be called back from G0 if we need to produce more liver cells, which is kind of cool. All right, the G2 checkpoint. This is controlled by DNA replication and completion, DNA mutations, and cell size. This is the checkpoint before it moves into mitosis. the G2 checkpoint. The M spindle or metaphase checkpoint is where we check spindle fiber or microtubule attachment to the sister chromosomes at their anchor sites called kinetochores. What do you think would happen if a cell tried to divide, but its spindle fibers weren't attached appropriately to all of the sister chromatids? What might you end up with? You could have some daughter cells that end up with both sister chromatids. What if they didn't separate? That would mean that another daughter cell wouldn't have any of that chromatid at all. All right, so at the G1 checkpoint, if a cell receives the go-ahead signal, then it moves on to the S phase. If it gets a red light, it'll go into G0 phase, and it won't divide. All right, the mitotic spindle checkpoint in metaphase. This is where we check to make sure that those spindle fibers are appropriately attached to each of the sister chromatids at their kinetochores. And you're zoomed right in, and you can see what the kinetochore looks like on a real cell. All right, internal regulatory molecules also play an important role in regulating these checkpoints. So if there's a fluctuation in a control regulatory molecule called MPF and cyclin, you can see how they change during the cell cycle. So both of these regulatory molecules surge in concentration during the mitotic phase. Kinases, like cyclin-dependent kinase, CDK, is a protein or an enzyme that controls the cell cycle. And it's active when it's connected to another one called cyclin. Cyclins are proteins which attach to the kinases to activate them, and the levels fluctuate in the cell cycle. Internal regulatory molecules. So MPF is the maturation promoting factor. It's specific cyclin CDK complex, which allows cells to pass G2 and go on to M phase. There are also external regulatory factors like anchorage dependence, density dependent inhibition. So this would be when if there's too many neighbors, cells won't divide. Anchorage dependence would be if a cell can't anchor, it won't divide. All right, and here's a good example. So on the left, we have density-dependent inhibition, where it's getting too crowded, so the cells stop dividing. This is normal behavior for mammalian cells. On the right, however, we see cancerous cells. And cancer is just uncontrolled cell division, uncontrolled mitosis, right? So here we see that density-dependent inhibition has been inactivated. These cells don't seem to care that there are way too many of them for that space. They just keep dividing anyways. All right, growth factors are proteins released by other cells to stimulate cell division. Density-dependent inhibition is when crowded cells stop dividing. Cell surface proteins bind to adjoining cells to inhibit growth. Then anchorage dependence, when cells must be attached to another cell or extracellular matrix in order to divide. So cancer is a disorder in which cells lose the ability to control their growth by not responding to regulation. It's a multi-step process, and it includes normally 5 to 7 genetic changes for a human for a cell to transform from a normal cell into a cancerous cell. They lose their anchorage dependency and their density-dependent regulation. A tumor is a mass of abnormal cells. A benign tumor is a lump of cells that remain at the original site. A malignant or invasive tumor impairs the function of one or more organs, and that's called cancer. Metastasis. This is what happens when cells separate from a tumor and travel to other parts of the body. For example, let's talk about breast cancer. So if a tumor grows from a single cancer cell, in a breast we have this little red tumor here. Those cancer cells invade neighboring tissues. And the cancer cells can spread through the lymph and blood vessels to other parts of the body. The cancer cells may survive and establish a new tumor in another part of the body. That's called metastatic tumor. A tumor that's begun to grow, but it's been seeded by another tumor somewhere else. All right, so what can be done to prevent cancer? Well, anyone could possibly get cancer, but there are some ways to minimize our risks. Don't smoke. And that includes hookahs, chew, and secondhand smoke. Use sun protection. Wear sunscreen. Don't get sunburned. Exercise and keep your weight at an ideal level for your body. Eat lots of healthy fruit and veggies per day. 5 to 7 servings of fruit is a lot, probably more than most of us get in a day. Use screening and preventative measures like breast or testicle or mole checks. And practice abstinence or use condoms. Some cancers are sexually transmitted, like cervical cancer. And get vaccinated. So luckily, cervical cancer, we actually have a vaccine to prevent cervical cancer, which is crazy cool. But cervical cancer is caused by the human papillomavirus, and you can be vaccinated against it. All right, so by now, you should be able to describe the events of the cell cycle, describe how mitotic cell division results in genetically identical daughter cells, and explain the cellular basis of cancer. All right, as always, if you have any questions, please write them down. Bring them with you to class or stop by office hours or attend Zoom hours so we can chat about it. All right.